The Buruli Ulcer Rapid Diagnostic Test, RDT, is intended to detect the active infections of Mycobacterium ulcerans, the causative pathogen of Buruli ulcers. The rapid test detects the presence of mycolactone, a cytotoxic metabolite of Mycobacterium ulcerans. This rapid test is formatted as a competitive assay. This means that the assay produces a strong test line in the absence of mycolactones and a weaker test line in the presence of mycolactones. The fact that this test line disappears in the presence of mycolactones may be counterintuitive, but is correct. Note that the intensity of the test line can be quantified with a visual scorecard. Place the test as close as you can to the visual scorecard and assign a number. To be considered positive, the test line intensity in the presence of mycolactones should be at least 3 shades weaker than in the negative control. One difficulty of working with Buruli ulcers is that the mycolactone is collected from the ulcer with a swab and is then very diluted. To overcome this problem, this assay uses innovative magnetic nanoparticles that can be concentrated with a magnet. Materials required: cassettes. These will come in individual sealed pouches. A vial of lyophilized magnetic nanoparticles. Extraction buffer. Deionized water for reconstitution of the nanoparticles and for swab extraction. A dropper with a chase buffer. 1.5 milliliter oxygen vials. 10 micron mycolactone in ethanol. Magnetic rack. Hydrofloc swabs. Pipettes. Pipette tips. 8 milliliter amber glass vials. Aluminum foil or dark box. A timer. Gloves and a vial of lyophilized HABA. We will go through all the steps in detail in a moment, but here's an overview. The HABA and lyophilized nanoparticles are reconstituted. Next, a swab from the lesion is dipped in the extraction buffer. In step 2, the HABA and lyophilized nanoparticles are added and incubated for 30 minutes. During this time, any mycolactone present in the sample will adhere to the nanoparticles. At this point, the vial is placed in a magnetic rack, and the nanoparticles accumulate on the walls of the vial as shown in step 4. The supernatant is discarded and replaced by 2 drops of chase buffer. The nanoparticles are reconstituted in this chase buffer and are transferred to the test. Now let's familiarize ourselves with the test by running a negative control and a positive control. First, a vial of nanoparticles are reconstituted by adding 300 microliters of deionized water, which is provided with the test, solubilized in nanoparticles for 5 to 10 minutes with gentle mixing, and make sure that all the material on the walls are solubilized. Each vial contains material enough to run 5 tests. Unused reconstituted nanoparticles can be stored at 4 degrees Celsius and used up to 2 weeks after reconstitution. Next, let us prepare the negative and positive controls. It's a good idea to run them in duplicate. To prepare the negative control, add 500 microliters of TEA extraction buffer to the vial. If you would like to ensure best results, you can add 1 to 2 microliters of absolute ethanol, but this is optional. To prepare the positive control, add 500 microliters of TEA extraction buffer to the vial. Add 1 to 2 microliters of mycolactone and mix well. This comes in a glass ampule and is very precious. Do not waste it. Add 40 microliters of magnetic nanoparticles to each vial with positive and negative controls, using a new pipette tip each time in order to not contaminate the magnetic nanoparticles. Shake manually or vortex briefly. Incubate the mixtures for 30 minutes by letting them sit on a regular rack while protecting them from light with aluminum foil or a dark box. Mycolactone is light sensitive. At this stage, keep the magnetic rack and any other magnets away to not interfere with the assay. Once 30 minutes has elapsed, place the vials on the magnetic rack for 30 minutes. Protect them from light. Cover with aluminum foil or a box. The nanoparticles will accumulate against the wall of the vial. Open and label the cassettes. The cassettes should be removed from the fridge 40 to 60 minutes before the start to allow them to come to room temperature. After 30 minutes on the magnetic rack, you will see the nanoparticles accumulate and form a tiny dot. Remove the liquid gently from the first vial with a pipette. We insist on the word gently. When decanting, place the pipette tip close to the wall opposite of the magnet to not disturb the particles. Wash the particles from the side wall of the vial and mix gently by pipetting several times. Do not be too vigorous. You do not want to create foam. Transfer the vial content to the sample port of the cassette. Start timer or record the start time. Repeat the nanoparticle collection for each sample. Read the tests after 10 minutes and again after 30 minutes from the start time. Bring the test as close as possible to the test card and score the test line intensity. Take a picture. As mentioned before, because of the competitive format, the assay produces a strong test line in the absence of mycolactone and a weak test line in the presence of mycolactone. In order to be considered positive, the test line intensity in the presence of mycolactone should be greater than or equal to 3 shades weaker than in the negative control. Now, let's run the test with a sample. In our case, we use a simulated sample. Before you start the test, reconstitute a vial of HABA in 0.5 milliliters of TEA extraction buffer. This is enough for 10 tests. Mix and let it reconstitute for 10 to 15 minutes. In the meantime, place the collection swab into 500 microliters of extraction buffer for 5 minutes. After 5 minutes, mix and then squeeze against the wall before removing the swab from the vial. Then add 50 microliters of the reconstituted HABA to the mix. Shake manually to combine. Then add 50 microliters of the reconstituted nanoparticles. You may have noticed that from the positive and negative controls, we are using only 40 microliters, while here we are recommending to use 50 microliters. The reason is other protein compounds in the extracted collection from Buruli wounds might interfere with the test, and the test line intensities might appear weaker than with the positive and negative controls. Then proceed with the test as before. Place the vial on a magnetic rack for 30 minutes, protected from light. The nanoparticles will accumulate against the wall of the vial, but this time it may not be as obvious as with the positive and negative control. Open and label the cassettes. After 30 minutes on the magnetic rack, remove the liquid gently, placing the pipette tip close to the wall opposite the magnets to not disturb the particles. Add 2 drops of chase buffer into the vial and remove the vial from the magnetic rack. Wash the particles from the side wall of the vial and mix by pipetting several times. Again, be gentle so you do not create foam. Transfer the vial contents to the sample port of the cassette. Start a timer or record the start time. Read the tests after 10 minutes and again after 30 minutes from the start time. Bring the test as close as possible to the test card and score the test line's intensity. Take a picture. Compare this test to the positive and negative controls.